As part of the introduction to microbiology, we will also be talking about the prokaryotic and the eukaryotic cell structure. By the way, before we continue, I have a disclaimer. We will not be able to tackle everything due to the limited time we have, so you will still have to read your textbooks, okay? But I hope we will be able to cover the most important things you need to know through our lecture series. Okay, so prokaryotes versus eukaryotes. Now, when we say prokaryotes, we will be referring to bacteria. Yes, archaea are also prokaryotes, but we will be focusing on bacteria for now. And for the eukaryotes, we'll be talking about the fungi, parasites, and even the human cells, just so we know the differences. So let's start from the genetic material going outward. So first, the chromosomes. Now, prokaryotic cells usually only have one chromosome, and it is in circular form. Eukaryotes, however, have multiple chromosomes, and the chromosomes are linear. Take note also that if you do find a prokaryotic cell with multiple chromosomes, most likely it is a growing cell about to divide. Prokaryotic cells have double-stranded chromosomes. Eukaryotes also have double-stranded chromosomes. So this is one area that both have similarities. Okay? The chromosomes of eukaryotes are wrapped in histones, while prokaryotes do not have histones. Eukaryotes have exons and introns. So let's suppose this is a long strand of double-stranded DNA, and this strand contains multiple genes. In eukaryotic cells, some parts of the gene are expressed, which we call exons. Expressed meaning their messenger RNA eventually gets translated into peptide sequences, while some are not expressed. So these genes that are not expressed are what we call introns. So they do not code for any amino acid. So eukaryotes have both exons and introns, while prokaryotes, maybe to maximize space, they only have exons. Now, because you have multiple chromosomes and more complex machinery in eukaryotes, you will need to have a nuclear membrane. Prokaryotes, on the other hand, do not need a nuclear membrane, so the prokaryon is just a clump of genetic material, and it doesn't have a clear nucleus. So that's why we call them like a nucleus, so nucleoid. Eukaryote cells have the presence of a nucleolus, which is a region in the nucleus where ribosomal RNA and proteins are synthesized and assembled. There is no nucleolus in prokaryotes because it has no nucleus in the first place, no? Okay, so another difference is the presence of ribosomes. Well, actually, both have ribosomes, but the ribosomes are in different forms. So please take note of this. This is very important. The ribosomes present in eukaryotes are 60S and 40S subunits. So one unit altogether is 80S. While in prokaryotes, they are in 50S and 30S, altogether 70S. Now take note that the S's here stand for Svedberg units. Okay? So it is a measure of a particular particle size based on its sedimentation rate. In other words, how fast a particle of a given size and shape settles to the bottom of a solution after centrifugation. So the ribosomes are made up of two different subunits. In eukaryotes, the first subunit has a sedimentation rate equivalent to 40S, and the other subunit has a sedimentation rate equivalent to 60S. But when they are centrifuged together as one unit, they have a sedimentation rate together of 80S. Alright, in prokaryotes, one subunit is 50S, the other is 30S, but when both subunits are centrifuged together as one unit, they have a sedimentation rate of 70S only. Okay, so take note that this is not an arithmetic problem, okay? So now, why is this important? Or why is it important to know this? Well, this is important when we go to pharmacology and we talk about certain antibiotics, okay? So it is important to know that bacterial ribosomes are different from our own cells' ribosomes. And so, our ribosomes are not affected by certain antibiotics whose mode of action interfere with the function of the 50S and 30S subunits of bacterial cells only, okay? And so, if if we come up with a drug that interferes with bacterial ribosomes and our own ribosomal functions, of course, that is not just a drug, that is a toxin. Alright, another difference is the presence of membrane-bound organelles. And by the way, in eukaryotic cells, including humans, not all DNA is in linear chromosomes. Okay, wait. All DNA within the nucleus are linear in form in eukaryotes, okay? But did you know that there are also DNA present in the cytoplasm of eukaryotes, including ours? Yes. It is present in, before that, it is believed that millions and millions of years back, some energy-producing bacteria entered into eukaryotic cells. And we were very glad to host them because they provided us with the energy while we provided them nutrition and support. And we developed a symbiotic relationship or endosymbiosis. Then maybe our cells decided to reproduce with them as part of its own, until now. But of course, we don't call them bacteria. We call them mitochondria. So like bacteria, they have their own genetic material. Have you heard of inherited mitochondrial genetic diseases? Okay, so this genetic material is a lot similar to those in prokaryotic cells. They are circular, singular, only exons, no introns, no histones, okay? So many organelles are present in eukaryotic cells. We already mentioned the mitochondria, right? So they are not present though in prokaryotic cells, no? If so, how do bacteria produce energy? How do you think? Remember, mitochondria are bacterioid organelles. So if the mechanisms for the respiratory chain are in the membrane of mitochondria, these are also present in the bacterial membrane, okay? Not in separate organelles in the prokaryotic cell. Because the prokaryotic cell do not have membrane-bound organelles, unlike the eukaryotic cells. Eukaryotic cells also have lysosomes, but lysosomes are not present in prokaryotic cells. The endoplasmic reticulum is present in eukaryotes, but not in prokaryotes. So, the ribosomes in the prokaryotes are actually free ribosomes that are floating around in the prokaryotic cytoplasm. What else? The Golgi apparatus, the peroxisomes, and cytoskeleton are structures that we can find in the eukaryotic cell. Okay, so what is in prokaryotic cytoplasm? So prokaryotic cytoplasm often store reserve materials in the form of insoluble granules, or we also call them inclusion bodies. So for example, glycogen, also sulfur. They also have large reserves of inorganic phosphate in the form of granules of polyphosphate. These granules are also sometimes termed as volutin granules or metachromatic granules because they stain red with blue dye, such as those in your Mycobacterium tuberculosis and your Corynebacterium, okay, which you will learn more of in later discussions. So take note that the most numerous intracellular structures in most bacteria are the ribosomes because, diba, again, they are free-floating around the cytoplasm of bacterial cells. Okay, so what else are different between the eukaryotic and prokaryotic cells? Okay, another difference is in the way that they divide. So prokaryotic cells have only one chromosome, right? So that one chromosome will simply replicate into two. And then the cell will then divide. A very simple way to divide. They fizz into binary bodies. That's why we call it binary fission. In eukaryotes, however, there are multiple chromosomes, right? So of course, each chromosome has to be duplicated first. But if you are going to have two daughter cells, you have to very carefully divide all the chromosomes in such a way that one copy of each chromosome should go to one daughter cell and the other copy goes to the other daughter cell. So in eukaryotes, we have to divide the chromosomes in a very specific fashion in the middle of the cell so that errors may be avoided as much as possible. So for that purpose, the cells make mitotic spindles. So it makes sense now, no? Before you were just memorizing the different stages of cell division. Now you appreciate why mitotic spindles are important. So these mitotic spindles attach to each chromosome and then divides the chromosomes appropriately and carefully. So amazing, no? Now in prokaryotic cells, do we need mitotic spindles for simple binary fission? No need na. So again, prokaryotes divide by binary fission and eukaryotes by mitosis. And of course, you are intelligent, no? You know that germ cells divide by meiosis. Except for some fungi, because while molds divide by mitosis, yeasts replicate by budding. Another difference is the presence of a cell wall in all bacteria except one. Okay, so which bacteria does not have a cell wall? Remember this always, okay? Mycoplasma do not have a cell wall. All other bacteria have a cell wall. Now, bacterial cell walls are typically made up of peptidoglycan. Don't worry, we will go into further detail about the cell wall in a while because it will be very important, especially when we correlate it with pharmacology. Now, why do prokaryotic cells need a cell wall? So this is important in prokaryotes because their cell membranes do not have the tensile strength to keep the cell intact. The prokaryotic cell is hyperosmotic. And if you remember your physiology, it becomes a magnet for water. And when water continuously enters the cell, it will undergo osmotic swelling. And if they keep on swelling, they are prone to osmotic burst. But because of the cell wall, the prokaryotic cell does not burst. Okay? But how about in mycoplasma who do not have a cell wall? So actually, their cell membrane, instead of a cell wall, the cell membrane of mycoplasma contains sterol, just like eukaryotic cells, just like our cells. So it is the sterol that makes the cell membrane tensile enough to prevent osmotic burst. Now, in eukaryotes, do eukaryotes have a cell wall? Of course, human cells do not have cell walls, no? But some eukaryotes have a cell wall, for example, fungi. But their cell wall is different from those of prokaryotes because they are not made of peptidoglycan. The cell wall of fungi are made up of chitin. Yes, the same protein in the shells of crabs and shrimps. Now, this is important to remember. Again, we are going to correlate it with pharmacology. So, we mentioned that bacterial cell walls are made up of peptidoglycan and fungal cell walls are made up of chitin. So, these are totally different structures. Now, we have certain antibiotics whose mode of action affects the peptidoglycan cell wall. Example, penicillin and cephalosporins. Okay? Now, because of that specific property, they are effective against the peptidoglycan cell wall, but they will not affect the cell wall of fungi. So that is why these antibiotics will not work against fungal infections. Alright? So if you prescribe these antibiotics to a patient with fungal infection, you will just be wasting the patient's money and you will be harming the patient by not actually addressing the fungal problem. Now, what do you think will be more effective against mycoplasma? Penicillin or cephalosporins? The correct answer is none of the above. Why? Because these antibiotic agents work only on the peptidoglycan cell wall, and mycoplasma do not even have a cell wall. Remember? So for this specific bacteria, we have to use something else. Alright? Now, we also mentioned earlier that eukaryotes have sterols in their cell membrane, right? Now, fungi also have sterols, but the sterol present in their cell membranes are ergosterol. And here's another pharmacorrelate. We also have certain drugs that affect ergosterol particularly. For example, amphotericin B. Now, this drug will be effective against fungal infections, but it is not going to affect the other eukaryotic cells whose cell membranes contain other forms of sterols, like for example, the human cells or our own cells. Why? What form of sterols are present in the human cell membranes? Cholesterol or cholesterol. Makes sense now? Alright. So if you notice, prokaryotic cells are simpler than eukaryotic cells at every level with one exception. the prokaryotic cell envelope is more complex, which we are going to discuss next.